All right, so now we have talked about some of the basic models, the Hardy-Weinberg model, how it's being used, can be used. And now what we're going to look at are this major part of the definition of population genetics, where we said it was really looking at the mechanisms that are responsible for gene pool change. We said all gene pools change. They're constantly changing. Some small changes over many years. Sometimes they're very large changes, depending on a number of variables. We want to start looking at some of those. So we're going to talk about what are the bases for modeling these mechanisms. Now just kind of bring us up to where we are. We're thinking about gene pools. We're thinking about gene pools changing. We're saying, yeah, but they're not supposed to change, right? Because there's this genetic equilibrium that occurs after one generation of random mating. So we keep getting pulled to the center, if you will, and then to get pushed, it takes a lot of effort. In fact, this equilibrium might be, in biological terms, equivalent of Newton's first law, the law of motion. Something placed in motion will remain in motion, or in the counter motion that comes from the other laws. But the point is, it's a principle, right? Hardy-Weinberg principle. We don't have many principles. We have the Mendelian principles, but as far as biology, as we said before, unlike physics and other things, we don't have this whole series of set hard rules. Things vary so much. But here's a basic principle that if we can study gene pool change and we can have an expectation of what will happen unless we have a very strong force acting. And just to bring us up here, across all of this is a big box that controls a lot of this and relates a lot of it, and that's environment. It can either be the cellular environment, the molecular environment, it can be the organismal within the organism itself, or it can, of course, be the external environment. This is where ecological genetics comes in, the relationship there, looking at how environment plays a role in changing gene pools. So, but for every gene pool, what's going to drive all these mechanisms is going to be some influence of environment. So what are those things? We said they were population size, this thing's mutation, drift, selection, migration, all play a role. So obviously, if you're thinking about a model of Hardy-Weinberg, each of these, in order for that equilibrium to hold, each of these must hold as an assumption. And the true hard assumptions of the Hardy-Weinberg is that you must have a large population, randomly mating, panmitic population. You can't have any of these other things going on. You can't have drift, you can't have selection, you can't have mutation, you can't have migration. Okay. Now, when it says no, that's a big word there. It turns out that you can violate all of these assumptions and still remain in Hardy-Weinberg. It's such a strong force that you can, in fact, be pulled back and be clearly in Hardy-Weinberg, even with selection, even with drift. But once you get it strong enough, or you get a combination of these things in play, then you can start to move away, as we saw in that example where we did the eggs from the Chiricahua Mountains, from the flies versus the adults. Well, obviously something had pushed that system away from an equilibrium. Okay? So in this particular lecture, I want to look at one big part of this up here, large, and then we'll talk about random mating, and then we'll move through these others. Okay. Now, large, and the reason I'm doing this is we could spend the whole semester on selection or drift. We've already talked, of course, about mutation, not in terms of a population, but of course. These are areas which you probably are not going to get as much exposure to, this idea of large and random mating, and as well as drift. So I want to cover it here because you get a lot of selection and other things many other places. But let's think about large, okay? Well, in reality, for Hardy-Weinberg to hold, the population has to be infinitely large. That's the mathematics of it. No populations, real populations, are infinitely large. They're all finite. There's always a set limit to how big or how many things you may have, okay? And that can fluctuate, of course, over time. Now, this leads to a major population question, which is what happens when you do have this finite size? What impact does this have on the gene pool? In other words, what does the deviation away from infinitely large have to do with finite? All right, so this leads to a major concept in population genetics known as the effective population size. It's a hypothetical construct. We can guesstimate it, we can estimate it, guesstimate it, we can try to figure it out. We can't count it directly, but we can figure it out. Now, some pedigree things you can work with it, but in real big populations, it's very difficult. But it is still an absolutely critical measure or concept that we use, and it's basically this. It's the number of independent genomic units successfully contributing to the gene pool of the next generation. Okay? So just how many genomic units contributed. That's what matters. That's what effective size is, the effective size, okay? And the effective size obviously is, is going to be different from the ecological size, the census size. In fact, anything that divides a population will normally reduce the effective size. I only know of one way that you can increase the effective size above the census size. That takes a little bit of work. Okay, but anything else almost always reduces the effective size. So what are some things that might divide a population? Well, we've talked about this before, separate sexes, right? That's one thing that can divide your gene pool, okay, dividing up into males and females. Obviously, age structure can divide it because it depends on who's contributing out of a particular group. And then finally, the last one is spatial structure that we'll talk about very briefly, okay? So sexes, remember that one? Remember when we talked about back in transmission genetics, we talked about sex linkage and all these things, and we said, we said, well, by the way, if you start to partition your genome into males and females, different functions happen to different males and females. In mammals, females are lactating, they're the ones carrying the young, so their gestation. There's different pressures than there are on the males. In birds, there can be, very similar, except the other direction. Sometimes you have color patterns and all sorts of things. But here we're talking about something else. Just think about it. You get these separate sexes. Does everybody get to contribute now to the gene pool? And if the answer is no, then that definitely affects the effective population size, and in fact lowers the effective size from the actual census size. One way is simple little way to calculate that, and there are much more complicated ways, but simple little way, the effective size can be the number, 4 times the number of females plus the number times the number of males, divided by the number of females plus the number of males, the total. This 4 comes from essentially meiosis, and we could do the derivations of this, but take my word for it. It's 4 times the number of females times the number of males. Let's take a real world example. Southern elephant seals were hunted to near extinction. A population persisted on an island, were found, and there on that island there were approximately 10 males and 40 females, so 50 total animals left. Let's pretend that all of these individuals mate. All 10 males get to mate with 1 or more females. Okay, what's the effective size? Well, it's 4 times the number of females, which is 40, times the number of males, which is 10, divided by the total. So in this case, the effective size is going to be something like 32, right? You say, well, that's not so bad, right? The next generation looks like it came from 32 animals instead of 50. Okay, we can work with that. But what about the real world? The real world, elephant seals have these harem systems, like many others, like white-tailed deer. These happen to be impala from Africa, but they have a harem system where genetically related individuals, usually sisters and aunts and cousins, are pretty much held as a group to protect themselves, and then there's usually 1 or a few males that mate with this group, right? So let's take an extreme case. Let's take these elephant seals and let's say that 1 of the males mated with all the other females. Okay, so now we've got 41 total animals. Okay, even if there are 10 there, genetically it's essentially a 41, even though there's 50 standing around. Okay, so what's the effective size here? Well, it's going to be 4 times the 40, but then just times 1, right? Because that's all it's going to contribute. Well, that's pretty much going to be somewhere around this pretty much all cancels out. We end up with around 4. Okay, so in this case, even though we had nearly the same number of animals, or in fact we could have had 50 animals but only 1 mated as a male, you have genetically about 4 genomes being transmitted. That's it. Okay? So it really can have a great effect, whether it's 2 males, 3 males, or something. You can play with the math of that. It's very trivial, and see where these numbers go. Now, it turns out that this little equation is the underlying model for a billion-dollar industry and has been critical for domestication for our entire time that we've done it. But it's worth $1 billion. So what is it? What's the idea? Well, the stud system. This idea that you take a male, and the male has characteristics that you want to transmit it as much as possible to the offspring. You mate that male with as many females as possible. The idea is you're trying to counterbalancing sexual selection as well as independent assortment, right? With the hope that you'll get as much of the genomic combination of this individual into one or more of its progeny that you can. Works really well for the entire stud system in horses and cattle and chickens and whatever you're using, whatever you're doing. Okay. Now it doesn't work so well in cases where you put them into captive situations and don't necessarily intend to do this. A good example is Przewalski's horses. These were horses that were nearly extinct in the wild. They're the ancestor of horses. The closest ancestral form is still alive, still persists. In steppes, China, and they were brought in. They were discovered in the 1800s. They were brought in, and only a few were brought in, obviously, and they used these stock with a very small number of males reproducing. And in fact, they maintained themselves separately in Munich and other places and in different zoos. Eventually, they allowed them to come together, but then we had wars, which broke all that apart, and we kept going back and forth. And what happened in these guys is that you ended up with a population over time that came from very few males that started the system. And this happens a lot in zoological parks and other places where, you bring in a few individuals, and originally, before we could transport like we can now, sperm and eggs around, we would just send a male around, right? This park would have a female. She need to be bred, so you just send this male, and this male might be responsible for half of the young that were born all over the world, that year or that country, right? And so you ended up doing the same thing. And now there's much more control over it. But still, you end up with things like these white tigers, right? That's the product of basically doing this in the wild. And it works fine if if you want to have a white tiger for everybody to go ooh and ah over. Unfortunately for the tigers, they have all sorts of problems. They have their heads are wrong. They have joint problems. They're just really, for them, it's not a very pleasant outcome. It's certainly beautiful outcomes. You want to see them, but they can't function in the wild, and they're certainly not wild animals. Okay, let's just do this one more time. Take it to an extreme case. Okay, let's just say an effective size of this. What if you had 10,000 females and only one male? No matter how many females you have, you asymptote about 4 genomes. So you got over 10,000 creatures out there, and you genetically are only putting 4 genomes in the next generation. Now, I know you say, well, when would that ever happen? That's not going to happen. Yeah, it happens in things like oyster beds, where you primarily have females and they're sexually reproducing, and they will take, and a female will convert to a male. They're broadcast fertilizer, so once the water is over the top of these beds, they'll throw up the sperm, and they'll fertilize many thousands of individuals. Well, genetically, that's very few genomes, even though there's maybe hundreds of thousands of individuals out here. If only a few of them are males, you've got a very small genetic variation that goes on in these groups, and it's well known that occurs, right? Right. Another thing that you can have is age structure, right? If you go to, here's some nice elephants wandering across Namibia, and here's young. Well, they're not reproducing at that point, so they don't count. Even though you count them as a census size, they don't count. And then it goes both ways, right? And if you, here's a little chart. If you were thinking about humans, well, there's pre-reproductive age, so those that haven't reached puberty yet, right? And that can be anywhere 10, 14, 15, 16. So these individuals at the bottom of the scale would be removed out at any one moment because they're not capable of contributing. And then in humans, we senesce. Our reproduction senesces, and certainly in females, by the time we get into our 50s, we hit menopause. So all of these females that are up here above 50 are no longer considered part of the pool. So it makes a big difference, right? If you are wandering around a college campus or a college town, you stand on the corner up here at Tumors, you count how many individuals go by and their ages, then you're likely to say that, well, there's the effective size is pretty close to the number of individuals I counted, because most of the people that you'll see wander by are reproductive age. But if you subtract the children that they may be pushing away, okay? If you go someplace, a retirement village, the villages in Florida or something, where I think you have to be 50 years old or something minimally to even to spend the night there. Well, if you count the population there, the effective size is pretty much 0 at that moment. Now they've all reproduced, so they've contributed, but at that moment, if you just counted them as living humans, their effective size is pretty low. The males are still functional, but most of the females probably have been reached menopausal age. Okay? So it plays a role. So you can subtract. Something else then is spatial substructure. Okay? Spatial substructure, if we divide up the populations over space, and this group over here doesn't, they're in a different valley than this one over here. They're not contributing very often, and that's going to reduce the effective size. Classic examples, if you go out to the western US and you see these huge herds in the winter of things like white-tailed deer or black-tailed deer or elk or things that come down out of the mountains. But when they actually go to breeding times, they disperse up into the mountains and up into these little valleys. And they also have harems. So you can actually have genetic issues because you go up and they're spatially subdivided. They're not moving around, and you end up with fairly small effective sizes of this whole population. So you might have 8,000 animals in the winter standing around in this area when they've come down, but by the time you go up, the effective size of that pool there may be in the hundreds or a thousand or something, much smaller than the actual census number. Last thing I thought I'd do is give you a real world example, something that I worked on, and just kind of show you what a role this can play. These are beach mice. They're found all along the Gulf Coast, starting over here about Fort Morgan, right there, running all the way down past Panama City here, various subspecies, most of which are endangered. Okay? And the most distinctive of them is the Santa Rosa beach mouse right here. It's not federally endangered, but the others are. They live traditionally on real dune habitats, that look something like this. They're one of the few small mammals that makes a full-time living out on those dunes because it's really a tough environment. Other things come out there frequently, but these actually live there full-time. They're monogamous, which is kind of cool for small mammals, and they live in burrows. The burrows look something like this. They're very stylized. In fact, there's a strong genetic relationship to how to make these burrows, but they'll dig down a meter or so, depending on the soil type. They'll dig a large nest burrow and then a small escape tunnel that'll go almost up to the surface, and they'll plug the entrance and with a sand plug. If that entrance gets broken while they're in there, then they'll scurry up this hole and dig their way out and escape. And again, it's a genetic trait. It's kind of cool genetic trait, right? So they use that. That's how they can persist on these frontal dunes, these northern natural areas. Now, normally, what happens is, yes, you get tropical storms come in or hurricanes and knock some of these back, but their population is big enough, it's back here on the back dunes, they repopulate and they kind of follow through the cycle of the whole things. All right, so hurricanes come along and they cause these sort of problems, right? Well, the problem now, of course, is that this is what the coast looks like. This is Destin, Florida, and this is what there used to be old beach mice, the Choctawhatchee beach mice, actually living right in here. I know they were because I've seen them. This is what it looked like when I was a kid. Okay, that's the same spot right there. Okay, different bridge, but this island out here, if you will, is sort of mostly human-made by dredging, is full of this now. There were mice here all along the coast, all the way down to Panama City, and there were even mice back in here. Well, obviously, they're not going to be back there anymore because they don't do well in parking lots or things. Okay? So what? Well, what is when you have a storm, and when it was natural like this, they would do fine. And even when you didn't have hurricanes, but just had winter storms, the water will come in and flood, but you still get enough populations. Now, hurricanes can produce all sorts of problems like this, right? This is right after a hurricane on the same beach. But ultimately, as long as they could move back and forth, you would have repopulation of these areas as the dunes return from this really destroyed state to this more natural state, which usually takes 5 to 8 years for that to happen. Okay? All right. Well, I was for many years worked on a task force that was trying to preserve the Alabama beach mouse, highly endangered. It had been a mouse that was found all along from Fort Morgan all the way up continuously over to Perdido Key. Here, not onto Perdido Key, but actually onto this little island that's found behind the Perdido Key, and it occupied that. Due to a development, due to all sorts of things through here, it was pretty much isolated down to a couple of areas, a couple of natural areas, Purdue Unit, which is part of the Bon Secours National Wildlife Refuge, and then down around Fort Morgan with some scattered individuals in between. All right, well, part of the task force thing, when you put species in the endangered species list, you have to come up with recovery plans, right? These recovery plans are how do we get it out of that status and back to maybe threatened, or can we ever get it back to off the list? And some things, bald eagles, for example, and alligators and things have come off the list, right, by careful management. So the question is here is, okay, what do we need to not necessarily get them off the list, just for them to persist, right? We were in this meeting and people said, well, how much space do we need for these mice, right? I mean, this land is incredibly valuable. How much are we going to preserve? And I said, well, let's try one of these little models. Let's say we want an effective size of 500. Not many mice. I mean, that's not even many elephants, okay? That's all we want. We want 500 Alabama beach mice, effective size, okay? Well, the first issue is that the populations aren't static. They go up and down and up and down and up and down, right? They do this in a couple of different ways. They do this both seasonally, because the breeding season's in the fall and winter, populations go up. They hit the summer, although we love to be on the beach in the summer. It's a really, really tough environment for these mice, so their populations decline, okay? And so then you have hurricanes hit, and that drives them even further. So even though you have these natural oscillations, these hurricanes can take them down to near extinction over time. But from our data, we said, okay, we basically have something where we can look at the number of mice that are out there, have some median number, and we can play with those numbers, okay? And so even though we have hurricanes and we have these nasty events, if we just figure out over time, from when there's a lot of mice out there, when the habitat looks like this, to when there's not many mice, when the habitat looks like that, we can figure out sort of a running number, okay? So I said, okay, from that number, we said, if we count up the number of mice that are out there and we think about the effective size, if the effective size to census size for most small mammals is around 0.25, okay, 25%. So about 25% of the mice that are out there actually contributing at any one time to the next gene pool. That's basically the way to look at it. Now that's a very liberal number. For small mammals, our real estimates are somewhere around 10%. Okay, so we were trying to be nice and say it's pretty large. Okay, all right. So in order to have 5,000 out there and considering all this oscillation, you need a running average of about 2,000 mice. Okay. Well, we put little radio collars on mice and followed them around and did kind of cool things with them, my students and technicians, and they found out that a mouse needs about 2,700 square meters of space to have what's called a home range, right? To be able to find enough food and mates and places to build burrows that they can persist in, right? If we take 2,000 times that, we get something like a little over 5 million square meters. To put that in some perspective, if we go about 250 meters deep, we really need to go about 500 meters deep, but if we just go back in the dunes a little ways, we need about 13.4 miles. Well, turned out to be, if we look at that, about the entire distance of Fort Morgan Peninsula, which at the time we were doing this analysis was worth billions and billions and billions of dollars, just tens of thousands of dollars per meter of beach. So this was, and of course it's critically important for the entire economy of the state because it's the small area of Alabama that's actually on the coast. So it's hugely popular. And we said, yeah, we need about the whole thing. Okay, give us the whole thing. That'll help us preserve the mice. But unfortunately, that wouldn't even do it. We said, yeah, that's true, but even if you had this whole thing, you really need to put separate populations because one hurricane might wipe this out. So you need some area over here and some area over here. And obviously that's just simply in today's conditions not feasible. But it gave us an idea, just using very simple numbers, these effective size, of what it was really going to take to try to preserve these. So it put that in people's mind. Yeah, we really, we don't have it. We're not even close. So we really need to think about other types of conservation measures that we can use, such as building berms and moving mice around manually and doing other things and preserve some habitat for them. So it was an important exercise based on simple numbers that we're using here. All right, so now we've done that. Now we want to come back in the next little lecture and talk about, okay, large is fine. Now what if they're not randomly mating? What does that do to us? Okay, that's the next issue that we want to deal with.